I'm thrilled to welcome Dr. Arlene Drack, who is joining us today from Iowa in the States. So Dr. Drack is going to talk to us today about gene therapy in BBS 10. Now, we wouldn't normally have a scientific presentation at the end of the day when our brain cells are flagging, but it is, I believe, is 8:45 in Iowa, Arlene?
Yes.
But this is exciting research and a fantastic way to finish our day. So thank you, and I'll hand over to you now.
Thank you. Can you hear me?
We can, yes.
Good, good. Well, thank you so much. It's so great to be here. I'm sorry that I couldn't hear all of the other talks, but it was deep night here in the US. So it's morning now, and I'm happy to be able to join you. And I'm so glad you recorded this. I'll watch the other talks later. So I want to start out by talking about a different kind of retinal degeneration called Leber congenital amaurosis. You see the eyes of this child here. She has nystagmus. The eyes have a sort of involuntary side-to-side movement that some people with BBS also get. And Leber congenital amaurosis causes a congenital inherited retinal disease. So BBS is a bit later onset. And in that way, it's a little different. But in other ways, it's very similar because these children also have a non-recordable electroretinogram. At the bottom of the screen, there's an example of an LCA patient electroretinogram on the left, and it's a flat line. And on the right is what one normal retina shows, which is kind of an upgoing bump. And this LCA condition is caused by more than 20 different genes. About 8% is caused by mutations in one gene called RPE65. And these are the visual fields of two different patients. On the top is a patient, the right eye OD is above and the left eye is below. And at the age of 6, the patient doesn't have good central visual acuity, but they have a pretty big visual field that's shown by the gray sort of circle. And you can see at age 12, it's a little bit smaller. At age 18, it's a little bit smaller. And by the time this person was in their 20s, they have only a 20-degree field, which is very difficult to be able to walk around and ambulate safely. Below that, there's a patient starting at age 11 where you can see the visual field, the gray oval, which is where they can see things, is pretty big. By the time they're 20, they have a little blind spot in the center and it's smaller. And by the time they're 28, they have like maybe a 5 to 6 degree visual field. And so that's the natural history of Leber congenital amaurosis due to RPE65 mutations. And that progression of visual fields is very similar to Bardet-Biedl syndrome, although the ages are a bit different. And the reason I'm telling you about this condition is because I was an investigator in the subretinal gene therapy trial for treating RPE65 retinal degeneration. And it was Children's Hospital of Philadelphia and the University of Iowa, and we enrolled patients between 3 and 42 years of age, and we treated both eyes. 31 patients were randomized for enrollment, and they were randomized 2-to-1 for treatment versus observation for a year. And I want to point out, as we kind of move toward clinical trials for things in BBS, that there are a lot of requirements that have to be met to do clinical trials. For example, in the US, the FDA required this randomization, which meant that 1 out of every 3 patients who were eligible for this trial could not be treated for a year. They had to wait for a year and have testing done during the year as a control group. Now, fortunately, they all were able to be treated at the end of that year. And the endpoint for this study was what we call the the multiluminance mobility test. And I'm showing a video here of one of the patients before treatment. And this patient was older than 10 at the age of treatment. And again, we didn't know whether treating at older ages would be beneficial because these people have very poor vision right from birth. And on the video, she is attempting to walk through this obstacle course, which has arrows on the floor that she's supposed to follow and obstacles, soft obstacles in the way. And if she is going off the course, then a helper gets her back on the course because that was the requirement for this study. But it's very difficult. She's really navigating by feel, which of course many people do when they don't have good vision. But a year after having this subretinal gene therapy, on the right is a video of her now really able to walk through the obstacle course very easily, following the arrows on the floor, going over obstacles. And this was really a massive, massive thing for me because this girl had a helper who walked with her through her school, guiding her to her different classrooms. And one day after this treatment, she was able to just get up and walk through the school on her own. So this was a huge high point in my career to see that this condition that I had never known would be able to be treated now had a treatment. And this is a graphic representation of how people did on that mobility test score. There's a solid line with circles that shows the people who were treated immediately. And 30 days after their treatment, they had an improvement in their mobility ability, and that lasted out to 4 years after the treatment. We're now getting later data. And the group with the dotted line and the squares, which is below it, are the people who did not get to have treatment in the first year because they were the control group. They had to do the same testing. So their testing did not improve over the first year. And then after they got treated a year later, 30 days later, their mobility improved, and they also maintained for 3 years on this graph. So this subretinal gene therapy for RPE65-associated Leber congenital amaurosis was called Luxturna, and it became approved for general use in the US in December of 2017 based on this phase 3 trial, and it's now approved to treat people ages 1 year and older who have this RPE65-associated Leber congenital amaurosis. And we're going to be following these patients who we treated in the clinical trial up to 15 years after treatment. So we know exactly how long the treatment lasts. Does it have to be repeated? Are there any complications, et cetera? And this, as I said, was really a high point of my career, and it's proof of concept that subretinal gene therapy can improve vision for people with inherited retinal degenerations. And BBS, Bardet-Biedl syndrome, is in that group of inherited retinal degenerations. So I have worked on Bardet-Biedl syndrome for quite a long time. This is a picture of the Bardet-Biedl Syndrome Family Association in the US. They had their annual meeting at the University of Iowa in 2014, and that was a really great thing for me to meet so many people and their families at one time. And you know, after the RPE65 Luxturna trial, we really thought that every retinal degeneration would really rapidly be treated or cured with this subretinal gene therapy. And we had all these animal models, and we started treating them. And there were also some clinical trials that started for different diseases. But what we found was it's going to be more complicated than that, because each disease works a little differently, and we have to really tailor the treatment to that disease. So all of you are very aware of what it's like to have Bardet-Biedl syndrome and what it's like to have the vision problems. This is from the ophthalmologist standpoint, what we see. On the left in green is an electroretinogram test, which measures the electrical activity of the retina from a patient who's 9 years old with Bardet-Biedl syndrome. And all of the recording lines are flat. They're just kind of straight lines with little squiggles, which are background electrical activity. And on the right in red is a normal electroretinogram from someone who doesn't have Bardet-Biedl syndrome. And there's a variety of different upgoing and downgoing waves depending on what kind of light we stimulate the eye with. And so this is from a patient with BBS 10 who was 9 years old at the time of this ERG. And he was actually seeing very well and functioning very normally. So the electroretinogram is a test that can tell us when there's a retinal degeneration present. It doesn't really correlate with visual acuity. And it's abnormal a long time before people realize that they have a significant vision problem. And the other important thing about this patient is that he had testing, genetic testing, and had 2 mutations in the BBS10 gene in trans. And what we mean in trans is we have 2 copies of the BBS genes, one from each parent. And in order to have BBS, you have to have 2 mutations in the same BBS gene. And there are many, many BBS genes, as you know. So in BBS10, this person had to have 2 mutations found to know that it was BBS10. And then we did parental testing to make sure that there was 1 mutation from each parent. And whenever there are clinical trials, you need that kind of very specific genetic testing to know if you're eligible for a specific clinical trial. Now, I'm showing you these visual fields from the same patient. When he was 9 years old, the visual fields were really normal, and his visual acuity was pretty normal. And then below that are his visual fields when he was 14, and they're very, very small visual fields now. And when the visual fields get to be around 20 degrees, that's when it gets to be difficult to walk around kind of without bumping into things. Even before that level, it can become difficult, but at that level, it's really, really challenging. And so this is very similar to Leber congenital amaurosis. And again, all of these similarities make me hopeful that we're going to have, in the future, similar success in treating BBS. So Val Sheffield at the University of Iowa found many of the BBS genes, and he also has made many mouse models. This particular BBS10 mouse model was from the comp repository, and he shared it with us. And on the right is a BBS mouse when it's a month old, and on the left is a normal, or what we call wild type mouse, when it's a month old. And the BBS mouse when it's a month old is actually much tinier than its siblings. And because of that, we have to feed them special food, and they have to stay with their mother longer, because otherwise they just don't grow up in a healthy way. Now, when they get older, they become obese, just like people with BBS do, for the same reason. They have this leptin resistance. And as we just heard from Dr. Forsyth, now there's a treatment for that, a really great treatment, setmelanotide. And I'm so glad to hear that it's come to the UK, because I have many patients who are on this. And I mean, not only for weight control, but just because they feel so much better being on this medication. They feel like it has improved their lives so much. So I'm really glad that you all will have access to that as well. So then we have to ask the question, does the eye in BBS mouse, or in the BBS10 mouse, have the same disease that people have with BBS10? And so these are pictures of an OCT. OCT is kind of like an ultrasound for the eye, but it uses light waves instead of sound waves. And so it shows us a black and white picture. And the important thing is it shows us all the layers of the retina. The retina is the lining of the eyeball. The eye is kind of like a hollow ball. And on the inside, it's lined by this very thin membrane that's maybe the thickness of cellophane or saran wrap. But in that little saran wrap layer, there are multiple sublayers. And the thickest bottom one, which is black in the OCT, is the photoreceptor layer. So in the wild type mouse, that thick black bottom layer is the rods and cones, and that's where vision really starts. And you can see that in the BBS10 mouse, even at 4 months old, it's there, but it's much thinner. And as we go to 5 months, 6 months, 7 months, in a wild type mouse, that photoreceptor layer or outer nuclear layer, that thick black band, stays the same size. But in the BBS10 mouse, it shrinks away. And by 7 months, we can't really see an outer nuclear layer or photoreceptor cells. And the same on the right hand is true with the electroretinogram. The BBS10 mouse has a low but recordable electroretinogram when it's 3 months old. By the time it's 6 months old, it's almost completely gone. So we were fortunate to receive a grant from Fighting Blindness Canada, and also funding from InVision 2020, and also funding from the Bardet-Biedl Syndrome Family Association to work on gene therapy in the BBS10 mouse. And this video I'm showing is of me doing a subretinal injection in a mouse eye. We use an operating microscope like I use for children, who have cataracts and things like that, but this is a very different procedure in the mouse. But if you can see it, sometimes it's difficult to notice, but there's kind of a bright white band that goes across there, and that is the retinal bleb where I've injected the gene therapy vector under the retina of the mouse. And on the right, you see an eye, a fundus of the mouse that has been treated, and that green area is green fluorescent protein. So in this one, we put a green fluorescent protein gene into the viral vector to see how much of the retina we're treating, and we could see that we treated about half of it. And I will say that we had started this work right before COVID, and when COVID shut everything down, they wanted us to get rid of all of our mice, and I had just treated a whole bunch of these mice, so we had to get special permission to not get rid of our mouse colony, and also the BBSFA gave us funding to keep these mice going. So then we did this swim assay, which is sort of like that mobility course in humans, because I found out the hard way mice will not walk through a maze, even though that they always talk about that, but they will swim. So in a wild type or normal mouse, if you put it in a pool of water, and it will look around and see a platform and swim to it. If the mouse cannot see well, then it takes a much longer time to find the platform. So we did a control study on normal wild type mice, and then on mice that have no eyes whatsoever due to mutation in the PAX6 gene. And we found that the mice with normal eyes and normal vision took about 3 to 5 seconds to find the platform. The mice who have no eyes take about 32 seconds. And so they still find it. They figure out ways to do it. They're, resourceful, just like humans, but it takes them a lot longer. So we can tell whether mice are using vision or not in this assay. And so when we have a BBS10 mouse that is untreated, it will swim around in circles and eventually find the platform, but it takes much longer, about 42 minutes or 42 seconds, sorry. But when we treat the BBS10 mice, they can really rapidly find a platform, like 3.92 seconds. So we know that it's making a big functional improvement. And when we do statistics on these times, we can see that the BBS10 mice that are treated can find the platform much faster than the untreated mice in both the light and the dark. But even more impressive is the electroretinogram. So the electroretinogram, as I mentioned, becomes non-recordable or flat even when people and mice have pretty good vision. It's an early indicator that there's something wrong with the retina. And in people with Luxturna treatment, even though they get big functional improvement, the ERG, the electroretinogram, never actually improves. But in BBS10 mice, the electroretinogram really improves markedly after gene therapy. So this, there's a picture of 2 tracings. One of them has a big positive peak, and then the other one just has little squiggly background noise. And the one with the big positive peak is the right eye of a mouse with BBS10 that was treated with subretinal gene therapy. And the left eye is the untreated eye. And the important thing here is these mice never have a light adapted cone ERG unless we treat them. So all of these mice only have this background noise. And then after we treat them, they develop this peak. And the same is true with this one that has 2 peaks below. This is the 5 Hertz flicker. It's just a cone response, the cells that see color. And these BBS10 mice never have a recordable 5 Hertz flicker until we treat them. So this isn't just like preventing degeneration, it's actually improving their retinal function. And this is the eye of a BBS10 mouse that was treated when it was alive. And then after it passed, we took this histologic section of its eye. And on the right side of the screen, you can see that all the normal retinal layers are there in different colors. And the red layer and the green layer is syntaxin 3. And the red layer is PRPH2, and those are in their normal position in those normal cell layers. Whereas on the other half of the eye, there's an entire layer missing. That outer nuclear layer is literally just like 1 layer thick or missing altogether. And you have lost a lot of those proteins that are supposed to localize in those layers. So we can tell this treatment works really well in mice. So how can we treat people with BBS10? And how old can we treat and still get an improvement like we see in the mice? Because these mice are treated pretty early at about 21 days of life. So we are not finished with these experiments, but we're looking at what stage are BBS patients at different ages that would correspond to when we've been treating the mice. And on the top of this slide is one of those OCTs. It's like an ultrasound, but with light, of a BBS10 patient's retina when they were 8 years old. And on the bottom is a child without BBS who we did that same test on. And the patient with BBS at 8 years old has about 36% of the thickness of that outer nuclear layer or photoreceptor layer as a child who doesn't have BBS. And so we looked at the mice and we found that when mice were about 3 months old, they had maybe 50% of the normal photoreceptor layer. And when they were 5 months old, they had about 30% of the normal photoreceptor layer. So when we're treating at like a 21-day-old mouse, that's probably like a young child, like maybe 2 or 3 years old. It's not exactly accurate to compare mice lifespan to human lifespan, but just a rough idea. When we treat at 3 months old, it's probably like a pre-adolescent child, like maybe 5 to 10 years old. And when we're treating at 5 months old, it's probably like a teenage child in the early years. So this is trying to see if we treat it at slightly later ages, so children and teenagers. And so we treated these mice at 3 and 5 years old, or 3 and 5 months old, and we looked at the electroretinogram and that visually guided swim assay. And for the 3-month-old treated mice, at every time point, the ERG amplitudes were higher in both the light and the dark. And at some time points, they were very statistically significant, especially in the dark. And in the swim assay, where the smaller bars, shorter bars mean a better result because that means it took them a shorter time to find the platform. At both doses of the vector that we used, these mice had better swim times, even when they were treated at 3 months old. And this lasted out till they were 11 months after treatment, which is really pretty old for a mouse. And the 5-month-old treated mice, as expected, didn't have quite as robust a response as the 3-month-old treated mice, but the purple bars on this graph are the ERG amplitudes of the treated 5-month-old mice, and the green are of untreated mice who were followed at the same time. And at every time point after treatment, the treated mice had higher ERG amplitudes than the untreated mice. And this is really, really impressive because it was totally possible that when we looked at treating these older mice, they would not have any improvement. But the fact that they do have better ERGs than the untreated mice is really, really hopeful that even at later ages, we will be able to get some effect, possibly. The other thing that's interesting is if you see there are these little dots way above the averages, and these are what we call super mice who just had phenomenal improvement after the gene therapy. And we see this in humans too, sometimes for reasons we don't understand. The majority of people in a clinical trial will get a kind of a mildly good effect, and then a few people will get an extremely good effect. So we're trying to figure out why do some have such a great effect compared to others. So in conclusion, mice with BBS-10 that we treated at 3 months old had significant improvement in rod function by both ERG and that visually guided swim assay. And mice that we treated at 5 months old definitely had a trend toward improved function on their ERG as well, which is really, really hopeful. And we're still following these mice and doing more experiments on them. And so our future work is we want to better correlate what the OCT thickness between humans and mice is. So we'll know better, like, are there different doses that we should give at different time course of the condition? Or, we want to learn more about how the mice and humans correlate. And part of this is we have a natural history study going on right now where we have 9 BBS-10 patients who we've enrolled, and then we're doing all these studies and then repeating them within a year to see how much they change over a pretty short period of time. So we have 3 people who have completed 2 of the visits. So we have 6 patients more who are going through this, and we'll have more information about how to follow people as they have gene therapy. So I thank everybody who's been in this natural history study. So this is proof of concept that we can treat and perhaps reverse some of the retinal degeneration in mice with BBS-10. So everybody, of course, wants to know, how do we get this to human trials? Well, the vector that we used in the mice has not been used in any human clinical trials yet. So we made a new vector with one that has been used in human clinical trials that we should be able to get approved much more easily. And then we have to make a GMP vector. GMP stands for good medical practice, which means it's a vector that is of the quality and sterility that could be used in humans. So we have already made that vector in collaboration with Mira GTX, and we're now doing safety studies on that. What we found is there's a certain promoter that's part of the vector. There's a variety of different promoters you can use with these vectors. And one of the promoters kind of makes it overexpress, and that can be toxic. The other promoter really doesn't do that. So we've narrowed it down to the promoter that we need to use. We are continuing to do the efficacy studies to see, okay, what dose should we use? And then we're finding the endpoints through that human study that we're going to look at in people who are treated. So we're getting closer to the next phase, which would be to do a pilot or a phase 1 study. And a pilot or phase 1 study has a small number of people in it, and it is just to make sure that it's safe. And then once that pilot or phase 1 study is done, then we can apply to have a study that could enroll many, many people. And once the studies are done and it's actually approved, like Luxturna is now, anyone with the condition that's genetically identified can have the treatment. So we are now treating people outside the age range of RPE 65 that were in the clinical trial, because now that it's approved, anybody with RPE 65 can be eligible for treatment. And so far, at least in the US, insurance has covered the gene therapy treatment. So I want to end just by thanking again all of the patients and families with BBS around the world. I know that all of you live with BBS every day, and it is an incredibly challenging condition. I have great admiration for all of you. I want you to just know that I live with BBS every day too, because I know so many of you, and I know that there is a treatment that looks like it will work in this mouse model, and I want to get it to people. So thank you for your attention.